You're listening to CDKL5 in color. I'm Marissa, and on this podcast, I'm joined by my co host Amanda, and sometimes some very special guests to talk all things CDKL5 deficiency disorder. It's a rare genetic condition caused by changes in the CDKL5 gene. Patients with this condition often experience profound developmental disabilities, difficult to control seizures that start in infancy, cortical visual impairment, and a variety of other health concerns. It's a challenging condition, but we know that by sharing our stories, we can raise awareness, provide support, and paint pictures of our community that can lead to greater understanding and meaningful change for families like ours. Let's jump right in. Hi, Amanda.
Hey, Marissa, how are you today?
I'm doing good. We have a guest today. Our guest is Ben Goult, and he is here to talk about a recent publication that's come out that he's done work with in his lab with his group about CDKL5. So welcome, Ben.
Thank you,
Welcome.
Marissa. Thank you, Amanda, for the invitation to come and talk. It's great.
We're excited to have you. You're going to be our first science guest. I'm really curious to see how our community is curious about what you have to say, but you had a publication come out. I saw it on LinkedIn, and it came with this really neat video. So tell us, how did you get involved in studying CDKL5?
So it's a long story because I work on a different set of proteins. I work on how our cells sense and respond to physical forces. So I'm actually work on these mechanosensitive proteins, they're called, because they can sense and respond to mechanical forces and instruct the cell how to behave. Maybe I'll go back a little step. So I'm actually a biochemistry researcher. I'm a professor of biochemistry at the University of Liverpool, and my lab works on trying to understand mechanical signaling. We want to understand how the cells respond to forces, how they convert that into signals. And one of the things we discovered was that there's a protein in all of our cells, in all animal cells, called talin. And that's what we work on. No one will have heard of talin yet, but hopefully we'll change that soon. But what we discovered in 2014 was that it's built of these regions called domains, which behave like switches. When you pull on it, they open, and when you let go, they close. So these switches turn on and off different cellular programs. So that's what I went to set up my research lab on, and that's what we work on. These binary switches, which are built into our brains, which switch on and off different processes. And one of the experiments which we did was to try and see what binds to these, what interacts with these different switches. And we found an enzyme called cyclin-dependent kinase 1, which is CDK1, and which is involved in how cells divide and in this process called mitosis. And from there, we looked at what other cyclin-dependent kinases also would interact with that domain. And we found that cyclin-dependent kinase like 5, CDKL5, also interacts with the 8th binary switch domain in talin. So that got us into understanding CDKL5 regulation. And then my PhD student, Yuzumi Otani from Japan, who's just submitted her thesis, and in that she took the CDKL5 project forwards. And what we noticed was that CDKL5's got a business end, like a kinase domain, which is what is the active part of the enzyme. And then it's got this really long unstructured region, like a piece of spaghetti. And at the very, very C-terminus, there's a highly conserved region, which Yuzumi noticed had the perfect sequence to be an anchor to interact with some of the scaffold proteins in the brain. So from that moment on, we suddenly knew that the very C-terminus of this protein, which everyone had kind of ignored, was anchoring the enzyme in the synapses, in the dendritic spines. And that immediately gave us, coupled with what we know about these binary switches in talin, a model for how CDKL5 might be being regulated, and also an explanation for why premature stop codons might be so damaging to the protein's function, because this protein is trying to anchor by its very last amino acids. So any premature stop codons cut that anchor off, with the unfortunate consequence that it then mislocalizes and floats away instead of being anchored. So I don't know if you will be able to share the video, but it's conceptualized quite nicely in that. And that video is made, it's a structural video. We made it from the actual protein structures. So it's all to scale, it's all accurate.
That's really interesting. We will be able to share the video. We'll link it in the show notes. So when you're doing this work, this is my lack of science knowledge coming out. How are you getting these spaghetti pieces? Are you getting them from animals, or are they mini brains, or how are you getting this stuff in your lab?
Um, so we don't get them from humans, and we've got the sequence. So some bits we make as a synthetic gene. We just order it from a company, and they print it. The full length constructs we ordered them from a really useful thing called AdGene, which is a for scientists. You can look to see who's worked on these proteins before, and then order that piece of DNA to test. One of the things we've had to do with regard to AdGene is that the sequences in AdGene are incorrect because they don't have the anchor region at the end. So Neil Ball, who's a postdoctoral researcher in my lab, rebuilt the CDKL5 plasmids so that they're correct now. So now we've put them back in. So hopefully, fingers crossed, all researchers will be able to use the correct neuronal version now, which has the anchor. Because up until our paper, unfortunately, it's been a lot of research has been done using constructs which don't have this anchor region. So we don't get it from brains, we get it from plasmids, and we're a biochemistry lab. So most of the work we do, we actually make these proteins in bacteria, in small little E. coli. We use them as little protein factories, and we make the individual bits of talin and the individual bits of CDKL5 so we can have pure protein, and then we can do experiments to see how they function, what mutations might do to them, characterize how tightly they bind, and all these different type of biochemical approaches to understand how the molecules work. And then based on that knowledge, we collaborate with a neuroscience group in Helsinki, with Juha Sarakangas, and he tested all our CDKL5 constructs in actual rat neurons, and showed that if we tag at the very C terminus, if we put a fluorescent tag on, because if you want to look at things in cells, you need to put a fluorescent tag on them really to be able to see where they are. Unfortunately, everyone's been putting the fluorescent tag at the C terminus, which gets rid of the anchor region. So we show in our paper that a tag at the C terminus, it's just everywhere. But if you tag at the N terminus, put the fluorescent protein at the very N terminus, then you get beautiful localization to dendritic spines and synapses in the rat neurons. And also what's interesting, I think, is if you have premature stop codons in CDKL5, you tend to have less synapses per dendrite, like you have a smaller number. But if you overexpress this N-terminally tagged version in the rats, they get an increase in spines, which would fit with this protein being important for maintaining and stabilizing the synaptic connections.
And the synaptic connections is what helps us with learning, right?
Yes, it does. And it also helps us with connecting all our inputs and outputs, like so that we can respond and move and coordinate our bodies in response to signals. So if they're unstable, then those connections don't form quite as accurately and quite as robustly, and that can cause some of the symptoms and some of the problems which occur with mutations in this protein.
I'll chime in and just say our kids are sensory. I've had my kid in compression garments. Like, beautiful to have you telling us that you're figuring some of this out today, because phenotypically, I do think that, the sensory piece is a big piece, and it could be a big piece of why some of the kids don't achieve motor milestones that others do. Proprioception is a big problem. So, very exciting.
Oh, thank you. Yes, hopefully we can make a bit of a progress on understanding this condition and working out ways to mitigate it or improve it. I don't know, but we understanding what's actually going on is the first step to trying to work out how you might improve life.
When you're doing your work on the talons
Yes.
and you come across something like CDKL5 and you know that it's a thing that's being studied, is that more exciting for you than to come across something that, a different series of letters and numbers that are a gene that nobody's talking about? Does it make a difference for you?
So yes, it does. And one of the things we've got very interested in, because we found these binary switches which are inside all of our synapses in our brain, we're very interested in trying to prove that there might be some sort of binary coding at play in our brains, which is quite science fiction-y, but these switches are essential for brain function. And if we take talin out, the brain doesn't work at all. You don't even form a brain. So yes, and linking talin to a really important neuronal enzyme like CDKL5, and then being able to see for the first time how CDKL5 might be being regulated, why it needs to have that really long unstructured region, which you'll see in the movie, because most proteins don't need a long unstructured region. But if it's serving as the tether when this thing's anchored to the membrane, it's like a buoy in the sea where it floats it's attached to the rocks beneath. That's exactly what CDKL5 is doing. It's attached to the membrane, and then it's got this unstructured region, and then the enzyme here which can move up and down. So tying that and then working it out and then seeing how talin was affecting that, yeah, it's very exciting. And the fact that we might be able to make a big difference to a very specific illness is also exciting.
Well, I think you've done a great job explaining your very complex work in a way that people are going to understand at the level they choose to, which some of it will be seeing that video and hearing you talk and being like, all right, we're moving forward. Because you're probably the first scientist I've heard speak that maybe has something going for the mechanism, right? The whole thing has been, we don't even know what this does. And when we tag things it doesn't make sense, I guess, is just to simplify it to my level of
Yes.
understanding. So I'm extremely curious where your research goes next. If you have anything more to say about where it currently is, I want you to finish that thought. But if you have some more to say about where you think this research needs to go to, help us get closer to drug development and things that impact daily living, I'd love to hear it. And no
Yeah.
pressure. If not.
Yeah, no, so these are all good questions. So in terms of our immediate research, the direction of travel where we're going with this is that CDKL5, everyone thinks about it just in the brain and its role in synaptic function, but it's also in most of our other cells as well. So we're looking at how it regulates other tissues as well, which is a bit more off-piece from the epilepsy, but might explain some of the motor or some of the other peripheral symptoms. So we're looking at how it controls talin and how talin controls CDKL5 in how cells move around in culture, to try and understand the basic what this enzyme is actually doing. Because the problem with a lot of these ones which cause a disease, because we also work on Alzheimer's disease and a protein called amyloid precursor protein, everyone focuses on the bit which causes the disease and not what it's doing in everyone else or what its main function is. So we really want to understand what CDKL5 is doing and this sort of mechanical ability to move and be regulated by mechanical signals. So we're very interested in that. The other thing which we're incredibly interested in is, as I said before, this protein's got a kinase domain, a long unstructured region, then an anchor. If you look at that unstructured region, which looks like spaghetti, if you looked at it in a model, you'll see it in my video. If you look at that region, there's 6 motifs, they're called, like 6 patches, which are super well conserved. So they are almost certainly binding sites for things we don't know what they do. And they've kind of been ignored for a while, I think. I've not seen a single paper on them. But if you look at CDKL5 when it's all scrunched up into a ball, then all of those conserved regions are really close. But if you stretch this out, then those conserved regions are at specific locations within the synapse. So we think that CDKL5 might also be positioning other molecules which might be contributing to the correct functioning. So one of the things we're really keen to do is, when we found CDK1 bound to talin, we did that by doing this thing called proteomics, where you pull talin out of a cell and then you send it for a technique called mass spectrometry, and it tells you what's stuck to talin, what's bound. So we're trying to do that same experiment at the minute with the CDKL5 unstructured region, where we pull it out of a cell or out of a neuron, and then we send it to the mass spec facility, and they'll tell us what else is bound to that. because it might be that things which are bound to those conserved regions, and they're incredibly well conserved, they're in every single animal going really far back. So whatever they do, evolution's kept them. So they're doing something important. And so we might find something which is interacting with CDKL5, which is treatable or druggable already with existing drugs. So we're very interested in that. So they're probably our two main lines of inquiry: what it's actually doing and what these motifs might be doing.
Fascinating. I mean, every part of our children's bodies are affected and impacted by this disease. So thank you for thinking about more than just the neurons in their brain, because I really think some of the quality of life and ADL things that are happening have less to do with the epilepsy than, whatever else is going on with the disorders.
Mm-hmm.
So very excited to, hear more in the next year or two about what you guys find.
Hopefully we will have some nice updates from those experiments I've outlined there. They look very promising. And also we put all the tools straight in the database. We're trying to add genes so everyone else can work with the correct versions. We're trying to update the rat gene so that everyone will use the correct versions, because at the very least that will accelerate, just because many people can all start working towards with this new information. And it's like with new information, it always seems obvious looking backwards, but until you find the new information, nobody is aware of it. So it's not like a slight on the field. It's just, I think we found something really significant here, which hopefully will accelerate progress in this area.
Yeah, absolutely. That is amazing. And, we do get hung up a lot on the epilepsy and CDKL5 because it is so relentless and it has such
Yeah.
an impact on our kids' lives. But there are a lot of challenges our kids face that live in conjunction with their epilepsy that are not necessarily because of their epilepsy, like Amanda said. And, improved motor function for our kids would be tremendous.
Yes.
Improved hand function. I mean, my son doesn't have head control, um, when he gets ill, he's in the hospital very frequently. Like just these other parts of life that impact, like Amanda said, quality of life.
Yeah.
Yes.
Any
The
small improvements in any of the symptoms that our kids have would
Well,
make a big difference for families'
Yes.
quality of life. Yeah.
the
Yes.
sensory piece, the stemming, vocal stemming, that yelling, head throwing, like it all.
Yeah.
When you talk about the mechanical aspect of things, that it's exciting to me. I'm not doing a very good job putting words to it, but some of my daughter's biggest challenges of having her out in the community is she could be very loud when she's overwhelmed. Light. There's all kind. Everything that I am assessing is what's the sensory profile of this place we're going, because it doesn't matter what drugs we give her. That can be a problem.
Yeah, no, that sounds incredibly tough. So I wouldn't want to get anyone who's listening's hopes up that we're close to having a treatment here.
No.
What I think we're close to having a detailed understanding of what this protein is doing in its normal form and its normal version, and that might point to new avenues for developing treatments to soften some of these conditions. So just to put it out there, because at the minute, like we've got a very basic understanding of this, but I think it'll go fast. And I think understanding the proteins at the protein level is key, because until you understand what the protein's actually doing, then trying to understand what it's doing in a neuron or what it's doing in the brain, it's like you can't really understand it. But by understanding what the protein does, then hopefully we can then understand it in the neuron and then in the brain, etc.
So I've one last question. Is proteomics kind of where AI has really catapulted science, or do you feel like it's kind of stood on its own aside from AI?
I think proteomics is just super useful because there's so much stuff in the cell. There's loads of proteins which nobody's ever worked on really, because they don't directly cause a disease, so there's all this unknown stuff in there. And AI is helping a bit to map interactions, but I would say it's still it hallucinates a lot. So you have to validate them all properly. But proteomics, you pull a load of stuff out. You might pull out 1,000 proteins. So you have to do a lot of careful controls, and then you might end up with 100 proteins which look promising. They're always in the experiment, never in the controls. But then 100 proteins is still a lot. So then you need to actually work out if you believe that's true. So that takes the time, and that's how we found CDK1. It was just stuck on the side of talin in all of our samples. And it took us down this amazing route into now working quite squarely in the neuroscience space, trying to understand these neuronal enzymes. But whether AI will help proteomics data analysis, but I think there's still a need for biochemists.
It's still a long road. I mean, I think that's kind of what I'm trying to help people understand is it takes it's time. You guys, it's a
Yes,
lot of work to tease
yes.
this stuff
And one
out
of the
in the
things
lab.
which has been very helpful in some regards is now AI and like AlphaFold, and they made a program called Alpha Missense, which can take every single possible variant in a gene and try and classify whether they think it would cause a condition or not. So that's great. But the big problem, and one of the things, we're working with quite a few children who have got mutations in talin-1, which in talin gene. And we can show from the family tree that these are either a de novo mutant or actually with a family history. Um, and we can show at the protein level, at the cell level, and at the patient level that this variant's linked to a disease state. But these classifiers at the minute don't classify those talin mutants as being pathogenic. And that's because talin, because it's got these switches, it's constantly it's changing its shape and it's changing its structure and what it's doing. So something which would be very subtle in a protein, which is just like a ball, can be really bad in a protein which is shape shifting and doing all of these different things. So I think, so we've been working with Los Angeles Children's Hospital and the Children's Hospital in Barcelona and Boston Children's Hospital on different variants of talin which cause different diseases, not just epilepsy but other things. And the first one we got working on was with Los Angeles Children's Hospital. And it was really interesting because he had his genetic report of this really, really unwell child. And at the very bottom, at the footnote, it said, we also note a de novo variant in the talin-1 gene, but talin-1 is not associated with any diseases as of whatever the date was. Check back in 2 years' time. So that was just written there and then ignored. But then we were looking at mutations in talin which cause cancer, and it happened that the same amino acid in talin, the codon which made it, had a mutation in the DNA. So when the doctor revisited that 2 years later, he searched for talin and that residue, and all of a sudden it came up. So he got in touch with us, and that took us down this path of like trying to understand how talin was linked to disease. But it leads to this idea that at the footnote of all of these genetic reports, there might be loads of these where talin's flagged but not linked. So I think as we learn more, we'll realize just how many of these very conservative mutations are called, where they only change something ever so slightly, can still be pathogenic. And I think that will be a big step forward in our understanding of talin in neuronal function.
Well, and that's the thing about these genetic tests, right? Is like you were saying, Amanda, about AI, and you still need people. These genetic tests, you still need doctors to put the clinical picture together with the mutation, because we have people that have CDKL5 mutations that don't have CDKL5 deficiency disorder because they don't meet a clinical picture of having it. Isn't that right, Amanda?
Yes, sure. Not every mutation is disease causing. Most mutations I see still, I mean, it's getting better, but most of them are considered VOUS, like uncertain significance, even though, but I also see these reports now to his point that have 10 different genes listed that could have a neuro effect. And, I'll have to start looking out for talin when people share it
Yeah.
with me. But, that's the thing too, is you send your DNA off and whatever they're sequencing, they decide what they're going to share with you. So I think it's always a good idea, especially if you've had like whole exome or genome sequencing, to check back with your testing, whatever, especially if it's an academic center, like check back every couple years if you're moving along through a disease and it's like, it doesn't feel like I fit in with these people, because it's very possible you don't. Or maybe you have, I think the idea that you can only have one thing wrong is, I think some of these, it's like, all right, well, I could see how you can have two different diseases as well. So I think that's the key, is we know a lot more about genetics than we did 20 years ago.
Yeah.
But there's so much more.
And I would say that's the thing, our knowledge and the sequencing data, it's just accelerating so amazingly fast. And for most proteins, that classification is pretty good. And if it's a mutation which doesn't change, like if a mutation adds a premature stop codon, that's pretty obviously that's really bad because you've stopped the protein early. But if it just changes one amino acid to another amino acid, and that amino acid isn't in a really important part of the molecule, that could be quite benign. It might not even be pathogenic at all. So being able to separate that, like find the ones which are likely to be bad, I think we're making huge progress on that, and AI is starting to come in on that. But I still think, yeah, as Amanda says, having the doctor to look and having the basic science to work out what these regions are doing is essential still. So it's used as a tool, not as a replacement for actual scientists.
Right. We need scientists. We love scientists. We're so
Yes.
we're
We need
so grateful.
biochemists.
We need all of you. We're so grateful that
Yes,
you are looking at CDKL5 within your work, and all the other researchers that are doing the same. And we do walk this line of being excited when people learn new information. And of course, as parents, we want to take that and be like, well, how's that going to help my kid? But there is a big gap. There's a gap of time between what people are learning in labs and what eventually might become something that helps our kids. And I think,
yeah,
as parents, we have to learn to reconcile that all of
Yeah.
this takes time. My son's 9, Amanda's daughter's 20. I remember when my son was first diagnosed, I thought, oh, well, there'll be gene therapy in a couple years. But you know what? Newly diagnosed families, I think, also have that. And you have to have that hope at the beginning.
yes,
Like, this is not going to be as bad as I think it's going to be, because it takes you a while to get to acceptance. Like, this is my child's life, and eventually get to a place where you're okay with that. And that's a journey that families have to go on. But initially, you're just desperate for some sort of hope. And
of course. Yeah,
um,
it's unimaginable, and that thought that there's hope. But then, if you saw on the news this week the gene therapy for Huntington's and stuff,
Huntington's.
like
Yes.
that's absolutely incredible. Like I read that, and you're like emotional, and it doesn't even affect me personally, but it's just this is a glimpse of there are opportunities and understanding how these things are working. Yeah, let's hope we can
Yeah.
make a
I mean,
difference.
you give me hope because forever it's been like, what does this gene even do? Right. I mean, at least we know that there's an anchor, there's a scaffolding, there's some things that are important that we've been missing, right? Like where that takes us is it can only be to better places, I guess. So a little bit better understanding because I
I
think
hope so, and I hope that yeah, understanding how it works and understanding what else is interacting with it, they might be more treatable than the actual disease or or CDKL5 itself. There might already be drugs which exist which interact with those, because it might be that if CDKL5 isn't anchored, it diffuses away. But if it's got all important things stuck to it, they might go away with it. So who knows? I mean, I'm not trying to second guess that we'll find something immediately, but I think there's a lot of leads we've got now and a lot of new insights. So yeah, hopefully, fingers crossed.
Yeah, that's awesome. And you're going to be at the upcoming CDKL5 Forum, which is the annual science meeting hosted by the Lulu Foundation. So you'll be
Yes,
able to share with the people there, and put your heads together with the other smart people who are going to be there. And
yes, that's
I got
like
all my fingers crossed. You guys keep figuring this out, because Lord knows I
yes.
don't know what's going on.
Because when my preprint came out, Dan Lavery, who's the scientific
yeah.
director at the Lulu Foundation, contacted me and said, "You should come and speak at this and educate and present this because it's quite impactful and everyone needs to know this." So yeah, I'll be in Boston in a few weeks' time.
That's awesome. I'm so excited.
Fantastic. Well, thank you for being with us today, for being our first scientist to come and chat with the community. And
Hopefully, with that pressure of being the first, I've done an okay job.
you've done a great job.
It's
You've
been
done
a
a great job.
fascinating
Yes.
chat. So yeah, thank you for having me.
All right. Thanks
Good.
very much, Ben.
Thank you.
Thanks, Ben.
Thanks,
All
Melissa.
right.
Thanks, Amanda.
Thanks for listening. We hope you'll follow us on social media at CDKL5 in color on Instagram, Facebook, and LinkedIn, and visit us online at www.cdkl5incolor.com. Have questions or a topic you'd like us to chat about? Would you like to be a guest on our podcast? Email us cdkl5incolor@gmail.com, and we'll be in touch. Until next time.